the title is Sketching Alternate Realities, an Introduction to Causal Inference in Genetic Studies. What I'll do is cover the two main frameworks for causal inference, which are essentially identical, but provide different ways of thinking about the problem. That of the potential outcomes framework, or also called the counterfactual framework, and the graphical framework using directed acyclic graphs, also called Bayesian networks or causal graphs. We'll sketch, have an outline of how to think about the causal inference problem, why it might be hard, how we can use the tools provided by these frameworks to think about the problems, and then go through few examples in genetics, which may be viewed as applications of these frameworks to the causal inference problem. So I have to preface everything by saying that I'm basically a novice at this. So I'm new to this, as perhaps many of you. So I want to approach it with due humility. So if anyone knows more about something, please chime in, and we'd be happy to hear what you have to say. It is a notoriously difficult problem, both philosophically and mathematically and conceptually, and it is something that a lot of very smart people have thought about for a really, really long time. And in this 45 minutes or so, we are going to really scratch the surface. I'm going to gloss over a lot of details, maybe a lot of some important details. I hope that we will be able to at least get a flavor of what the problem is and how current approaches are approaching this problem. So as I said, the agenda is first to express causal inference as a missing data problem. That's the potential outcomes framework. Give an idea of what are the assumptions that are needed for causal inference. Express causal information as graphs, and then see how we can use those graphs to guide our analytic strategy. If you have any questions or comments, you may use the chat box if you know how to use Zoom, and either Laura or David will be monitoring the chat, and we can discuss issues or questions as they arise. So let's think about some of the most simplest possible causal inference questions. You have a drug that you want to know whether or not it affects an outcome of interest. And so here, let's look at a scenario here. There are 8 individuals, and I have a diuretic, that's my drug or treatment, and I want to see whether it affects blood pressure or not, systolic blood pressure measured in millimeters of mercury. So in the counterfactual framework, the way to think about it is that each person has 2 potential outcomes. When we are asking, does this diuretic cause the blood pressure of an individual to go down or change, we are asking the question, we gave this individual a drug, what would have happened if we had not done it? So you want to compare what happened with what would have happened. So in the counterfactual framework, or a potential outcomes framework, every person has two numbers associated with it. There is one number as corresponding to what would have happened if you had just given them a placebo, nothing basically, versus what would happen if you give them a diuretic. And the difference between the two is the effect or the causal effect of that drug. And so here, this is basically a cartoon of 8 individuals with two potential outcomes, one under placebo, one under the diuretic, which is our treatment, and the third column is the effect. And notice that the effect varies by person. Some people have a more effect than others. And the goal is to estimate the causal effect, or more accurately, the average causal effect of the diuretic versus the placebo in these 8 individuals. Now, notice that in real life, we can only give the person either one of these, either the placebo or the diuretic. We don't get to observe both of these numbers. We get to see only one. So in reality, it will more likely look like this, that if individual 1 gets the diuretic, individual 2 gets the placebo, individual 3 gets the diuretic, and so on. So basically, we get to see only half of the potential of the half of the outcomes that we want, that we need to know in order to estimate the causal effect of the diuretic. So immediately you can see that in order to estimate the causal effect, you have to impute the data in some way. And as anybody who has done imputation knows, in order to make an imputation, you have to make some assumptions, whatever they might be. And some people might be uncomfortable making those assumptions, or at least you would want to be cautious or thoughtful about what assumptions you need to make in order to fill in this missing data. So that's where the assumptions underlying causal inference come into play, because in essence, you are filling in the missing data. In fact, in the statistical literature, the causal inference problem was framed as a missing data problem, and a lot of the theory has come out of that way of thinking about the problem. Now, now some of you might say that, well, in certain situations, it is possible to have both placebo, give the individual both placebo and diuretic. In fact, you have these so-called N of 1 trials where you could have a crossover design where you randomize the order in which individuals receive the placebo and the diuretic. You would have to assume it doesn't come free. In that case, you would have to assume something about any crossover effects, and you would want to see if there are any effects of giving one before the other, and so on, any order effects. So even if you can give both drugs to an individual, you would still have to make some assumptions. And in many problems, you don't have the option. So for example, if the intervention is not a drug that easily washes away, like a diuretic, but something more substantial like a heart transplant, yeah, you cannot give a person a heart transplant and not give them a heart transplant. That's not possible. Or there are certain outcomes, like if the outcome was mortality, if a person dies, you cannot give them the other intervention. So it's not feasible. So in a large majority of the cases, we have to resort to some assumptions in order to estimate the causal effect. So now let's think about what we would need in order to get an estimate of the average causal effect. So in this cartoon example that I'm showing you here, the average causal effect is about decreasing the blood pressure by about 10 millimeters of mercury. So, well, one thing you could think about doing is you randomly give one person the placebo, another person, you choose who to give placebo and who to give the diuretic at random. So if you do that, then you are basically taking a random sample from this column, and by construction, since you're giving the diuretic to the others, you're also taking a random sample from this column. So in other words, if you go through this process, this is iteration, this is one instance of your trial, this is another instance of your trial, this is another instance of your trial, and so on average, your estimate of this column will be the average effect of the placebo, and this would be the average effect of the diuretic, and the difference between the two would be exactly what you need to know. So that is the central idea behind randomized clinical trials, where what you do is you're choosing who to give the placebo or the drug, the diuretic, at random, and that because that process is random, you can estimate the average effect of the placebo, the average effect of the diuretic, and you can take the difference. So essentially, the idea is that since you have to impute the missing data, you need to add some assumptions. And the more variables are involved, the more missing data you have to essentially impute, and increasing the problem difficulty. So, even with just one intervention and one outcome, you have half of the data being missing. Imagine if you have more variables, then you have more and more data that is missing, so the problem gets harder. So some of these assumptions can be rather strong, but if you have the ability to intervene, like as with a randomized trial, then you can estimate the average causal effect. And note that even with this, this way of randomizing, I can estimate the average of this column, average of that column, and take the difference to get the average causal effect. But I still don't have have a way, or a good way, to estimate the individual level effects. Again, without further assumptions, that becomes a true imputation problem, so which requires even stronger assumptions. So in other words, it's easier to estimate the average causal effect than it is to get the individual causal effects, which would be essentially doing an imputation using statistical models, which may require more assumptions than what I would have to make if I were doing just a randomized trial. Now, the average causal effect, as I said, can be estimated if we can assume that the treatment assignment was independent of the potential outcomes. Now, which is what would be if I was choosing who to give which treatment at random. But note, I want to show you this table again. Notice if I knew um, all of their potential outcomes, if I was like as it's commonly called sometimes, the perfect doctor. If I was the perfect doctor, I had this hidden ability to know what's best for every individual, I could give every individual the best possible treatment. And in fact, actually, uh, this assignment is basically the best possible outcome for every individual. You can see for one, the last one, both are the same, so you can pick any one. For the other one, individual 7, this is the placebo is better for them because that's lower. Um, for individual 6, everybody, almost everybody else except the first one, the diuretic is better. And so you can see that this is close to uh, the best possible outcomes for everyone. So this assignment, although it so happens I got this randomly, um, this particular assignment is actually not uh, is although it happened by chance, uh, and it can happen by chance in a small sample like this. In a large sample, that would be very unlikely to happen. And uh, but this particular assignment is correlated with the potential outcomes. It is designed in a particular way. So, in other words, the average causal effect can be estimated well only if we can assume that the treatment assignment was independent of the potential outcomes. If I knew what was better for everybody, then I would get a biased estimate of the effect of the drug because I'm choosing what's best for every individual. So I don't necessarily get the unbiased estimate of the overall effect of the drug involved. So another thing I need to assume is, I need to assume what's called no interference between units, which says that the potential outcomes of a unit does not depend on the other unit's treatment values. That is, if um, you know I get a drug, it get the diuretic, then it should not affect the outcomes of another individual who may or may not have received the drug. Of course, there are other issues like selection bias, non-compliance, differential dropout, measurement error. All of these things can affect the way in which can introduce bias into randomized trials and a study of this type. In a genetic context, the treatment or drug is like the gene, and the outcome is the disease of interest. And I want to come back to these issues. So in many situations, especially if you're doing a genetic cross, you can assume that the assignment of the gene or the allele is being randomized approximately, independent of the outcome uh, for that individual. There could be potential confounding by ancestry or family structure. That is, that would mean that some of the outcomes are associated with with the gene. So that could potentially be an issue. There could be selection effects. if only fit individuals survive or make it through make it out of the womb. And there could be potential interference if there is competition between littermates, and that the source of that competition is related to the gene of that we are interested in. For example, if there is a body weight or a body size locus, it is possible that if there is limits on resources, then the fact that an individual has increased body size due to the gene could have an effect on other individuals who do or do not have the same gene. So here is another scenario, the confounding scenario by ancestry. As I was mentioning earlier, there is a gene of interest that we want to know whether it is causing a disease of interest. And one of the things that we have to be careful about is whether there is some confounding due to shared ancestry or genetic relatedness or familial relatedness. I'll show you a famous example of a haplotype that was found to be associated with diabetes in Pima Indians. And the haplotype was present in, when the haplotype was present, the prevalence of diabetes was about 8%. And when it was absent, it had a 30% prevalence. So this appeared to indicate that this haplotype, GM haplotype, was protective towards for diabetes. But the wrinkle was this haplotype frequency depended on Pima ancestry, and the diabetes prevalence also depended on the Pima Indian ancestry. So in this case, the gene assignment, whether or not you would get the disease, depended on potential diabetes status. So this is the source of confounding, and this is why if we look at it straight like this, we might not get an unbiased estimate of the effect of the haplotype, which appears to be protective here. So the authors actually did an adjusted analysis and they adjusted both by age and by proportion Pima Indian heritage. And when you stratify by that, then you can see that there is virtually no difference in diabetes prevalence when you stratify by the amount of ancestry that an individual has. So that this is a classic case of confounding due to genetic relatedness. So we covered what the potential outcomes framework does for us. The basic idea being that the potential outcomes framework makes it clear that in order to assign cause, we have to impute data in some way. And we need to make quite a lot of assumptions if we want to estimate the causal effects for every individual. But if we want to estimate the average causal effect across individuals in the population, then we can get away with some weaker assumptions, even though, and they are worth thinking about, such as the no interference assumption and the ability and to ensure that the causal variable that you're interested in should be segregating independent of the potential outcomes that you're interested in. So now I'll go into the directed acyclic graphs framework, which is a very intuitive and very elegant at the same time way to express causal information. So the term directed acyclic graphs is the mathematical term. We are using it here to express causal information. So maybe we can call it causal graphs. These are also known as Bayesian networks, belief networks, decision networks, and so on. So here is a classic example of a lot of EQTL studies will have, at least implicitly, this framework. There's a gene of interest that causes the disease, you think, and you think the action of the gene is in part mediated through the expression of that gene or another gene, which in turn causes the disease. So that would be the framework that would be expressed in terms of this graph. Each arrow indicates, so this is one way to express the causal information, that gene is acting through the expression of that or another gene in order to cause the disease. There are different terminology that's used for directed acyclic graphs, and maybe I think the best word is probably causal, phrase is probably causal graphs. The mathematical term is directed acyclic graphs. They're also called Bayesian networks, but if you are a statistician or even a mathematician, you might be thrown off by the terminology. Actually, there's nothing Bayesian per se about the framework. It is really a statement about conditional probabilities. But okay, more on that in a second. So the way to read directed acyclic graphs or DAGs is, first let's explain the terminology. The word directed is there because the edges, these lines, imply the direction of causality. They are called acyclics because if you follow the arrows, you should not come back to yourself, the same point back. So that's why acyclic. The nodes or vertices of the graph are variables, observed or unobserved, and the edges arrows are the causal effects, as I said. One assumption that is made in order to make these graphs causal graphs is what's called the causal Markov assumption. That is to say, which is saying that conditional on its direct causes, a variable is independent of any variables for which it is not a cause. And this assumption allows you to write out the joint distribution of all the variables involved as a product over the nodes. Basically, you write the joint distribution as the product of conditional distribution of each variable conditional on all its causes. And this framework is completely non-parametric, as the conditional distributions can be arbitrary, and in fact these pieces can be learned by machine learning, statistical learning models, if as the case might be. So it's a very general, even though it looks just like a picture, but it's a very general framework for expressing causal information, and also gives you a way to write the joint distribution of all the variables. So now let's look at a few consequences of this. So first is that, if you have a graph, you can write the joint distribution. But if you have the joint distribution, you cannot necessarily infer the graph. I'll give a very, very simple example. I mean, it's almost tautological. Consider this graph here. So according to this graph, the joint probability of both x and y, x and y are two things, should be the product for over all the items, which is X and Y. For the first Y, it is the only cause is X. So it should be probability of Y given X. It should be, sorry, there should be a parenthesis here. Parenthesis times the uh, the probability of X, because X has no cause. So it's just that. But that, using, I guess, using Bayes' theorem here, can be written as probability of Y times probability of X given Y, which is consistent with this causal graph. So in other words, if I have the joint distribution of two variables, I have no way of telling whether this graph or that graph is the true model. So this is why, there's the saying, correlation does not imply causation. I cannot infer the causal direction by if I just have a sample of two variables from the population. And this is very general, very non-parametric. X and Y can be very complicated variables, and you can fit very complex models for probability of Y given X using machine learning, statistical learning, whatever else, but you still cannot infer the direction of the cause without additional assumptions or without additional information. So, I think this is there in some circles, there is the idea that with enough information, you can know what is causing what. The reality is that even for a very simple situation, that's not the case. You need additional information in order to put the arrows between variables. So this is almost a cartoon. This is, maybe not very convincing to somebody, but maybe should be also very convincing because it's very simple, very general. So here is another causal graph, and this is the causal graph corresponding to a QTL study. Those of you who have been to some of our other lectures may have seen a version of this. So here, in a QTL study, I am crossing two or more strains of an organism, and I genotype everybody because the meiosis in the process of crossing is scrambling the genotypes of the parents of the cross. And I am interested, if I'm looking at a particular location, I'm interested in knowing whether that locus is causing the phenotype. I am interested in knowing about this arrow here. Now notice in this graph, if I look at this graph, the phenotype is only caused by the QTL. The QTL is only caused by the meiosis, that is the state of the QTL is caused by the meiosis. But the meiosis also affect other markers on the same chromosome. So there's an arrow from the meiosis to the marker. So using this and the principle that I outlined earlier, I can now write the joint distribution of all four of these things. For brevity, I'm going to use some notation. The phenotype is Y, the QTL is Q, the markers are on that chromosome are M, and the meiosis is C. Then the probability of Y, Q, M, and C, all these four things, is the probability of Y given Q, because Y, the phenotype is only caused by the QTL, okay? Probability of Q given C, which is because the QTL is only caused by the meiosis, and probability of M given C, and there's a probability of C here, I forgot about that. So that is, marker is only caused by the meiosis, and so on. So that's this here, times probability of C, I forgot about that. So if you integrate or average over C, then you have this term, probability of y, q, and m is probability of y given q times probability of q and m. That's what happens when you average over, integrate over, or average over c here. And that can be written as, using just the law of conditional probability, as probability of y given q times probability of, this is the probability of the phenotype given the QTL times the probability of the QTL given the marker. And this decomposition tells you that you can treat the conditional on the QTL, you can treat the genetic model, which is the phenotype given the QTL, independent of the linkage model, which is the distribution of the QTL given the observed markers. This is this decomposition allows you to kind of separate the problem of kind of filling in the missing genotypes in a or unobserved state of an individual at a given locus separately from the problem of modeling the genotype phenotype model, the QTL genotype model. So this is just to show that you can use the causal model to come up with expressions that can simplify your analytic strategy or guide your analytic strategy. And almost all QTL analysis is based on this decomposition, whether explicitly or implicitly. So here, let's now go into a few other examples of how to read these diagrams. And this is a much longer topic, and you can look at some of the references later. So a path is a link from one node to another that goes through one or more nodes. A path is causal if it consists entirely of edges with their arrows pointing in the same direction. Kind of sounds obvious. Otherwise, it's non-causal, or it is association. So, for example, the meiosis is causing the state of the QTL, and so the meiosis to phenotype is a causal arrow, but the QTL and marker are associated. They are not necessarily causing, or you can say that they are causing each other. The other point I would like to make, and this may not be entirely obvious, but the DAG framework is equivalent to the potential outcome framework. It's been shown. So it's just different ways of thinking about the problem, and just some things are easier to see in one framework versus the other. Although their proponents might have they have disagreements, but at least from our vantage point, the points of their disagreement are probably finer than what most of us would care about. So this is the example of confounding by ancestry. Here, there is a causal link between gene and disease, and there is a confounding of the relationship between gene and disease that is confounded by ancestry. This is not a causal link. This is a associative link. So in other words, like if this link was absent, I might still see an association that is not causal. Here is two unlinked QTL. Here, this would suggest that these two QTL have independent effect, and but if you But if you condition on the phenotype, then you might see an association between these two. But that I'll come to a little later. I'll skip these in the interest of time. We have seen this expression also here. This is a mediator example. You want to see how much of the effect of the gene on the disease is mediated through altering the expression of a certain gene versus a gene having a direct effect on the disease. Um, so the other interesting thing that you can do with causal diagrams is to figure out conditional independence. And we've done a little bit already using the decomposition of the graphs. One notation we would need is that the idea of a collider. A collider is a node where two or more arrows collide. So for example, here, the phenotype is a collider because or here, the effects of the two QTL are colliding on the phenotype. A path is blocked if and only if it contains a non-collider that has been conditioned on, or it contains a collider that has not been conditioned on and has no descendants that have been conditioned on. This is a mouthful. But the main thing to note is that the unblocked path implies association. A blocked path implies conditional independence. So let me give you an example here. So if I condition on expression here, then it would block this link between gene and disease. And so then and then I would be, so if this let's say this direction was absent, that is, the gene did not have a direct effect on the disease. If I condition on expression, then the effect of the gene would be independent of the disease or uncorrelated with the disease, conditional on the expression. However, if we have this scenario, so here the phenotype is a collider because there are two phenotypes that are colliding, the two arrows that are colliding. Normally QTL1 and QTL2 are uncorrelated if we don't condition. But if we condition, then this path becomes open. That is, it can flow through the collider, and then QTL1 and QTL2 will be correlated with each other. So that's what it means. So let me show you another example, and this will be easier to see and easier to relate to. So this is an example of a asthma study, an asthma actually case control study, where there is a genotype of interest, and a lot of different information is collected on on these individuals, including obesity, and there are other covariates like sex, age, on which asthma depends, and which can affect obesity, which can affect asthma. The same what is of interest to us is whether the genotype that affects asthma also affects obesity or not, if there's this causal link or not, and if there is, how to estimate it. So let me give you so now I also told you this is an asthma case control study. So asthma is a collider, right? So because there are arrows converging on it. And so if this arrow wasn't there, let's imagine this arrow is not there. If I condition on asthma, which is what I would do if I have a case control study, because a case control study is done by selecting individuals who have the asthma and some individuals who don't have asthma. So it's by definition conditioning on asthma. that would open up this path between genotype and obesity through by conditioning on asthma. And so even if this arrow wasn't there, there would be an apparent association between genotype and obesity. So this is a classic problem of analyzing secondary phenotypes in a case control study. And I'll show you a picture to show you how that happens. So the idea is if we have a case control study and if we are interested in the association between a secondary phenotype, obesity, and a risk factor, such as genotype, that estimate will be biased. And I'll show you using a picture. Again, this is a cartoon. So I have uh, two phenotypes, a secondary phenotype. I just made it both of, and a risk factor. I made both of those quantitative, and they are actually uncorrelated. You can see this cloud of points. They are uncorrelated in the population, but they are both associated with the phenotype. So the individuals with asthma are in pink here. And you can see if you have higher levels of the risk factor, you're more likely to have the disease. If you have high levels of the secondary phenotype, you are more likely to have the disease. If I look at just people with asthma, you will see an apparent negative correlation between the secondary phenotype and the risk factor, even though in the population there is none. So if I condition on on asthma, that's how I induce an association between a secondary phenotype, obesity, and a risk factor genotype. The final thing I want to quickly cover, and I have a couple minutes here, I might go 5 minutes over, is instrumental variable estimation. And here the classic instrument is, of course, a randomized trial. A variable is called an instrument for a potential cause if it is only related to the outcome via the potential cause. And I'll show you an example. The best example is the randomization device that you use for clinical trials, because that randomization is what causes you to get the drug or the placebo, and it is only related to the outcome via whether you got the disease, the drug or the placebo. There are other examples like the draft lottery for exposure to war would be the risk factor, or and mortality or health outcomes would be the potential outcomes. In genetics and epidemiology, the polygenic risk score can be used as an instrument. And so here's the example of a polygenic risk score, how it works, and a causal diagram that helps you understand what happens, also known as Medelian randomization. So the idea behind the polygenic risk score is you have a bunch of genes that are used to come up with a polygenic risk score. That's a number. And that for predicting the levels of, say, cholesterol, that's your exposure of interest. And your outcome of interest is heart disease, which could be caused by other potential confounders that can affect this relationship between cholesterol and heart disease. But what happens is this polygenic risk score acts as your randomization device. That's your instrument. And if you are able to assume, and this is the thing, that these genes are affecting cholesterol through the polygenic risk score only, and don't have a direct effect on the heart disease, then this would actually help you get rid of the effect of these unobserved confounders. So that's why the polygenic risk scores are so attractive. Now, there are some assumptions involved, especially if you believe in a omnigenic model where everything affects everybody. This is probably not quite right, but it may be close enough for you to make make inferences of causality of the effect of cholesterol on heart disease. So in summary, the potential outcomes framework shows you how you can make causal inferences by framing it as a missing data problem. And that clarifies that you need some assumptions in order to make those inferences. Um, you need either assumptions or devices or interventions that allow you to fulfill some assumptions to infer causal effects. Directed acyclic graphs can be used to express causal information and can be used to evaluate what variables may be associated and what to condition on in your analysis. And this framework is very general, non-parametric, and additional assumptions can be made in order to improve information. Quick note on further reading and references. Most of the material in this presentation was gleaned from this book by Hernan and Robbins, Causal Inference. I think it can be enjoyed at different levels, and Miguel Hernan is a great writer. So any paper with his name, I totally recommend. It's been a very difficult field to understand, and I think he has made a major contribution in actually explaining things well. Another great book is Judea Pearl's Book of Why. Rob really likes it, Rob Williams. And there is also this nice paper by Greenland, Pearl, and Robbins. These are the people who have been behind much of this work on causal diagrams. I used the package DAGs to draw the figures that you saw in this presentation. The diabetes American Indian confounding example is from Nowler et al. And my paper with Gary is the source for the decomposition of the QTL problem. There are many other books, and in references. I would be happy to talk more about it if you have any questions or comments. And thank you for listening.
Thank you, Saunak. So it is officially 12:04, or at least in Colorado, it's 12:04. We're 4 minutes after. There aren't any specific questions in the chat unless Jeremy has a secret code of this 401532. But are there any questions from the audience or discussion items that they'd like to bring up?
So I guess I have Saunak, I joined late, but a question about how to add in the genotypes is something you and Shuhita and I have struggled with in formulating causal models. And I'm just wondering, I know we talked about the ability to model single SNP, one SNP, two SNPs, three SNPs, and 1, 2, or 3 genes. Is that sort of the limit? Do you think we're just limited to a very small number of genes in this kind of modeling?
We can't hear you.
The more variables you have, the more complex the system, more potentially assumptions you have to make. I think the genes are probably well structured, because we understand how genes work, segregate reasonably well. I think the major issue is the other things like, if you have expression or other intermediate phenotype, when are they measured? Are, you know, does cholesterol, cause heart disease, or heart disease cause cholesterol? You know, those are the, if they are measured simultaneously, that's very difficult unless you can make, you know, other ways to kind of deconvolve the situation. And so, yeah, I think that's like it is in the upper level above the genome, I think, where the complexity is.
Okay.
And the other thing is another thing that will come up, and it's not always transparent, is that if you are trying to infer the network from the observed data, uh, the more variables there are, the more candidate networks with, with support there will be, and that support might be diffuse, spread across graphs that look similar. How do you then combine, how, kind of in the information across those things? So that's where I think another place where it might get tricky. Um, that said, I think, I think with a manageable number of variables where you can think cleanly, I think, you can resolve certain errors, or at least you can use it as a hypothesis generating tool, which you can then use for further experiments to nail down something.
Mm-hmm. Thanks.
The polygenic risk score, that's really popular nowadays, especially with human geneticists. And I know that Laura and others, probably several others here in the audience, may be working on things related to that.
Could you walk through this one more time? I think I was struggling a bit with this one, and I think this one's really important.
So we are interested in the causal effect of cholesterol on heart disease. And the problem is that there are unobserved confounders that are causing both cholesterol and heart disease. So if we just have a sample from the population, this is a difficult problem because of these unobserved confounders. If there were known confounders, I could condition on it and get rid of this route that could cause the bias of this effect, but I cannot. So what I can do is, sort of, the idea is that, what if I could randomize cholesterol? If I had a way to randomize cholesterol to individuals, then I would have a better way of estimating the relationship between cholesterol and the effect of cholesterol on heart disease. And in fact, actually, for a continuous potential cause, the formula is covariance of the outcome and the instrument divided by the covariance of the treatment and the instrument. So imagine the instrument is randomization, then this covariance is exactly 1. So it's basically the covariance between the outcome and your cholesterol. That's your effect, So, but okay, now, but I cannot really randomize, right? What I have is a polygenic risk score. The polygenic risk score is constructed out of, maybe here it is 3, but it could be hundreds of genes. And together, it could be a reasonably good predictor of the risk of, good predictor of cholesterol. Okay, risk score, forget the risk score, but it's really, think of it as a predictor of cholesterol that you can learn maybe with a lot of data, outside data. So And the important assumption is that you have to assume that the effect of these genes is going through here. It cannot go like that, you know, cannot be. So, and that is an assumption. Because if you believe in so-called polygenic, omnigenic, like, Jonathan Pritchard, everything affects everybody. That would imply it's false, but maybe it is approximately true. Or the majority of the genes don't have an effect. Whatever is going into the predictor for cholesterol. And what you put is at the bottom here. Now, if it is a weak instrument, which means that the covariance is close to 0, then you can see that this will blow up. This will be a very unstable estimate. But if it is close to 1, then it will behave like a coin flip, like a randomization device, which will be a very good estimate of the effect of cholesterol on heart disease. So that's the basic idea, is to come up with a randomization device for cholesterol using these genes. And the stronger the instrument or this randomization device, the better you can kind of randomize cholesterol using these genes. Again, assuming you have to make assumptions like these genes are all approximately random, segregating randomly. Then you can have an effect of you can estimate a causal effect of cholesterol on heart disease.
The one that's really fun in this regard, Laura, is the alpha-5 receptor associated with increased nicotine use. Also confers a high intrinsic risk for breast cancer, for lung cancer, completely independent of its role in CNS. So just having the alpha-5 allele, I think, increases your risk for lung cancer, even if you don't smoke, almost twofold. So that's an exception where it would be a dicey gene to include in a model, because it would not be an independent factor. So in that case, it gets a little trickier, because now you have to tease apart kind of the relative weighting of the direct effect on, say, lung cancer susceptibility of alpha-5 and the mediative effect through, say, mesocortical limbic system, which is the one that actually is the major driver of lung cancer through its behavioral impact. So it's Judea Pearl has a long song and dance on this, which I think is, I found it a real eye opener, I think, in his chapter 9 of Book of Why. He actually has a lot of medically relevant examples here. He does talk about Medelian randomization, but he puts a lot of it in the context of Fisher and the battle about whether smoking caused cancer. So definitely well worth reading. So Saunak, in terms of directed acyclic graphs, so I mentioned that the Pearl book really doesn't, he doesn't even consider that explicitly. There's one section in the book where he kind of fesses up that real biology and real processes are often recurrent. And I'm wondering, is there some kind of idiot's guide to how do we reconcile these directed acyclic graphs with the ugly reality of biochemical pathways and intermediate metabolism and what's going on in the brain, where we know that there's just a ton of recurrence and feedback?
This may be outside my expertise. What I can venture to say is, if you break it down, that is, if you make the variables time dependent, then perhaps that's a way to make the circle into a line. If let's say there is a system where there is a feedback, if you make it explicit that the first, it's cholesterol at time 0, ejection fraction at time 0, cholesterol at time 2, ejection fraction at time 2, and put those arrows, then maybe you can make the circle into something that looks like a acyclic graph. That's what would be my thought here. But maybe the other people have other clever ways. But you know, you can think of, the graph is what is also a mathematical nicety, you know, right? It allows you to write this decomposition, this kind of like Markov assumption, because of the flow of information is in one direction. It allows you to write this product, and that's mathematically very powerful, very convenient, and allows you to do a lot of things. Yeah, so, but that would be my thought, but maybe others have other ideas here.
So Saunak, this is Bert Sharp. I think if you look at the endocrine system, feedback is not, negative feedback is not a discontinuous process as you're suggesting it might be when you say you could break it into time-dependent components or stages. It's actually a continuous process, because whatever we call the basal level is always acting on the forward movement of secretion, for example, and synthesis. And so really feed forward and feedback are in continuous interaction with each other in the system.
You know, you're absolutely right. I was just suggesting an approximation to the circular loop using a series of discrete time events. You know, you can theoretically make those smaller and smaller, but you're absolutely right that it would be an approximation. It is not modeling a true feedback loop. And you know, really, all it means is that this framework doesn't directly apply to that situation. You can make it fit that situation if you're willing to make some compromises, such as breaking it up into smaller time bins and trying to model it that way. But you know, it's like, that's not the truth. We are trying to use this mathematical framework to get an approximation to the truth, just like any model. So that's but you know, there are very elegant mathematical models of feedback loops. You know, you know, you can show what will happen to a system if you know, make it act a certain way. You know, you could think of our these SIR epidemic models. That is a feedback loop. You know that you could think of it that way, and there are all kinds of other systems that can be modeled. But that's a different kind of modeling.
Yeah, I think that Bert's comment is a really good one. And it does highlight the difficulty of syncing up the language and the thought process in our different communities. So, for example, as a geneticist, I tend to regard things as static systems. I have a genome, and long-term it will be associated with this phenotype. So I don't really consider it part of a dynamic biochemical network. And then you have people who are neuroscientists or cell biologists and are very, you know, used to the flux of enzymes and substrates and feedback loops. And it's really a different world with a different vocabulary and different assumptions about what we mean when we talk about a model. And so there is this collision between the kind of real world cell biological dynamic models and the statistical models, which do require, as Saunak really pointed out very well, a series of simplifying assumptions, but they're very powerful. If you can make those simplifying assumptions for example, in my world and in Saunak and Laura's world, the typical simplifying assumption is central dogma. Gene variants control phenotypes. And we don't really worry about the dynamics of that, right? We just say, okay, if you have this variant of this gene, you have high risk, and we can causally explain your risk, a certain fraction of your risk variance. And we don't have to invoke anything in terms of dynamic models, really. And that's a very powerful and actually true claim, but it's not satisfying to somebody like Bert who wants to know exactly how does that work, right? Which neuron in the shell of nucleus accumbens is doing it? And so bringing these worlds together, we're not there yet. And I think we're going to need huge sample sizes to bring those worlds together.
Yeah, and different ways of thinking. I think there are things happening at different scales, time scales, scales of matter. So I would expect that all the effect sizes that we are estimating today will be wrong 50 years later when our environment has changed in 50 years. Yeah, it's true.
When our diets are even worse.
Yeah.
That one thing we probably need to orient ourselves to, perhaps not at the moment, but over the time span of 50 years, is to think differently from the idea of a line. We probably should be thinking about a field of variables that interact, and the way, of course, the interactions are changing, and the field is quite dynamic. And models that can incorporate that idea rather than singularities that are implied in lines. I don't really know how to do that, of course.
You're
I think
definitely
that
Saunak's language, Bert, because
some
he's
of the,
Mr. Teacher.
obviously some of the tools are emerging with extremely powerful supercomputing that can look at fields of interaction, that can look at how molecules interact and predict the various ways that bonding can take place, right? And the probabilities of the different means of interaction. I mean, those are sort of like fields that are being defined, right? The many different vectors that lead to the interaction between a ligand and its receptor, for example, rather than just one line that defines the interaction, right? There are many different lines that define it with different probabilities.
Yeah.
Yes, I mean that what you're talking about is a bit like climate modeling, where with the physics at some level is very well understood, but that there are all these interactions between various components of the system is very dynamic. So I think it's a bit like that. Would you agree?
Yes.
So what they do right now is a mixture of like physics-driven modeling and which is combined with the empirical observations. So they, so perhaps something similar can be done for bio. I think people are already starting to do it, but maybe more of that kind of work will be done.
The amazing thing that I learned, and I can't wrap my head around it because I'm not a strong enough statistician. I'm not a statistician at all, is that Judea-Pearl claims that the nonlinearity is not a huge problem for the structural causal modeling. That is something I would love to understand at some point, how he overcomes the linearity constraints that we typically think of as kind of embedded in structural equation models and other approaches.
So he's right in that, like, if you look at, like this slide, is this slide still visible? So if you look
Yeah.
at this is just totally general. Is a graph is a statement about conditional probabilities, which can be anything. It has nothing, it can be arbitrary, you can use, and each of these conditional distributions, you can use very complex models. There's like no linearity. It could be deep learning, it could be logistic regression, linear regression, whatever works for you. In order to, if you have to empirically estimate each of these component causes. So in that sense, yes, no parametric assumptions are needed. But there are details, how the data arises, because ultimately it boils down to being, if you go to this this example, the simple example of this estimating the average causal effect, it is easier to estimate the marginal means here and take the difference if I have this randomization device. If I don't have that randomization device, and I want to, or even if I do have that randomization device, if I want to estimate the individual effects for every individual, I have to make all the full-blown assumptions that are necessary for imputation. So I think, to be honest, I don't understand everything either. Like, if you read any of these books, they will express an idea and then there will be a technical appendix that is impossible to read. So, but I think if I see it something like this: if you want to estimate the average causal effect, easy. If you have a device. If you don't have a device, then you have to make all these assumptions. And that's when, like, you can imagine, if you make parametric assumptions, you can fill in the data. We do imputation all the time. Actually, something like this, you could impute if, you would need a few with both observations here, but you can impute with some assumptions. But they would, assumptions means models, parametric, whatever, So that's that is at least my way of understanding it. But the other thing is that some of the disagreements between these people are things that don't matter to you and me, So